Despite intense research, the rates of allergic asthma is still increasing. We have developed a new idea to use specifically designed nanocarriers as inhalated vaccines. With this approach, the patients would benefit from much less medications and the healthcare costs would decrease significantly. To carry out this challenging project, we need an interdisciplinary team. And our team consists of Christophe Fongarnier, a pneumologist, Alke Fink, a material scientist, Fabian Blanc, a cell biologist, and me with a cell biology and nanotoxicology background. Christophe Fongarnier works at the University Hospital in Bern and he treats asthma patients every day.
Asthma is an extremely common condition affecting about 250 million persons worldwide. A great deal of them, about half, will have a significant allergy associated according to WHO statistics. Asthma is a chronic inflammatory disease of the airways that on the one hand needs an appropriate genetic background, but on the other hand has a multitude of triggers such as repetitive viral infections or exposure to allergens. Currently, the mainstay of asthma treatment is anti-inflammatory, and that means a lifelong inhalation of corticosteroids that will dampen inflammation in the airways in order to avoid chronic changes and chronic inflammation in these patients. And there's also a symptomatic component of the treatment that can provide dilatation of the airways as needed.
Alke Fink is working at the Adolphe Merkle Institute of the University of Fribourg, and she's developing new synthetic nanomaterials.
We synthesize and derivatize inorganic nanocarriers. We mostly work with iron oxide nanocarriers, since those are used already in the clinics as contrast agents for MRI. These nanocarriers measure around 10 nanometers in diameter. They are dispersed in a liquid, for example water, and they're highly magnetic. And we use this magnetism to actually fix these nanocarriers in this what is called magnetic reactor. Therefore, we load this magnetic reactor with a system of pumps and valves. In here, you're going to find a lot of permanent magnets that immobilize these nanocarriers in this magnetic reactor. In a second step, we apply all the chemicals we want to surface functionalize on such carriers. We take the magnetic field away, which means that we are releasing these nanocarriers, which then in the next step will be evaluated biologically.
I am working at the Adolphe Merkle Institute, and together with one of the two PhD students, we are focusing on nanoparticle cell interactions using cell culture models. The human lung with its huge surface offers the ideal target for inhalable nanocarriers. However, before we use newly developed nanocarriers for animal models or human exposure, we need some sophisticated cell culture models to study basic mechanisms. We have developed a triple cell co-culture model, which is composed of epithelial cells, shown here in red, macrophages, which are professional phagocytotic cells, shown here in light blue, and dendritic cells, which are the most immunocompetent cells in the human lung and important for the development of allergic asthma, in yellow. In combination with a liquid exposure system, which allows us to nebulize nanocarrier solutions over the lung surface, we have the ideal system to study nanocarrier uptake into cells and induction of cellular responses. Fabian Blanc and the second PhD student work in Christophe Fongarnier's research group, and they are working with animal models.
Animal modeling allows us to study the direct effects of nanocarrier on the whole organism. For instance, we are able to check into which compartments of the respiratory tract the nanoparticles translocate and with which cells of the immune system they interact in order to trigger a specific immune response. Nanocarrier tracking is done quantitatively using flow cytometry. Additionally, we are able to localize even the smallest nanoparticles in the tissue using high-resolution confocal microscopy. The most important advantage is the possibility to mimic a specific disease. That's why we established a mouse model of allergic asthma in our laboratory in order to study the direct effects of the nanocarrier on this disease.
Further systematic investigations are needed to understand the interaction of nanocarriers with the pulmonary immune system. First, very promising results have shown that nanocarriers have the potential to be used as inhalative vaccines to treat allergic asthma.
For me, it's absolutely essential in this project that the patient is in the center. And what we'd like to do is to reprogram the immune system in such a way that the asthmatic patient does not need any lifelong treatment anymore. This would also have a favorable impact on healthcare systems by reducing costs and simplifying asthma management.